This procedure begins with preparing an array of ACP1-GFP by first growing a lawn of cells and then pin out the array on a fresh plate. At the same time, prepare a fresh copy of DMA. Next, we mate the ACP1-GFP array to the DMA and select diploids, which are then sporulated on sporulation media. 5 days later, the meiotic progenies are germinated on LRK media on which only MAT-A haploids will grow. Through 2 additional rounds of selection, we recover haploid cells carrying both ACP1-GFP and a gene knocked out on kanamycin. Finally, we can pick individual strains directly from the plate and observe the mitochondrial morphology by confocal microscopy.
Hi, I am Shabnam Tavassoli from the Loewen lab from the Department of Cell and Developmental Biology at the Life Science Institute of the University of British Columbia.
Hi, I'm Chao, also from the Loewen lab.
Today we will show you a high-throughput method for incorporating organelle-GFP markers in various mutant backgrounds in yeast using a robotic system.
We use this procedure in our laboratory to study the morphology of organelles such as the endoplasmic reticulum and mitochondria.
So let's get started.
For this protocol, we use 2 different yeast strains. ACP1-GFP is a strain with a C-terminal GFP tagging of ACP1.